So good morning, everybody. And today we are going to carry on with our discussion about biomolecules, very basic introduction just to the molecules that make up biomass, the tissues that make up all living things. It's really important that we have some basic understanding. You'll hear words now that you've certainly heard before, protein, etc. This gives you some real idea about how those things are constructed. So I remind you, we talked before about how the carbon atom is the basis of all biological molecules. It may have other elements attached to it, but we are usually looking at a carbon skeleton for the biomolecule. And I remind you that we did this last time, that these carbon skeletons can vary in many, many ways. They can vary by length. They can put in double bonds, which causes the skeleton to be bent in space, not only in one direction, by the way, but also in 3 dimensions. We can have, we can put branchings into the skeleton, and we can also construct rings out of the skeleton. So those are the 4 major ways in which the carbon skeleton can be manipulated. And remember, in any one molecule, you may combine many of these there. So a skeleton may be very long. It may have multiple double bonds in it, and it may have multiple branches. And some of those branches may have rings attached to them, etc., etc. Gives a tremendous plasticity to the skeletons, which are made of the carbon atom. Okay, so we've already done that. Okay, so when we look at living things, we find that there are 4 classes of macromolecule. The macromolecule is a large molecule, and most of our molecules in living things are actually really, really big. So they are macromolecules. And the 4 classes are carbohydrates, lipids, proteins, and nucleic acid. So we're going to see very briefly some of the basic structure and function of these different macromolecules. Macromolecules may be so large they may contain thousands of covalently bonded atoms. They can be extremely large. In fact, one of the largest molecules in any cell is the DNA molecule itself, the hereditary material, because it has to contain such a huge amount of information. It is often very, 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 very long. Any one of our cells, we have nearly a meter of DNA carefully packaged to fit inside the cell. But if you were to string it out, it would be nearly a meter in length. And so that gives you an idea. One thing I want to emphasize is that the function of any molecule is intimately connected with its molecular structure. I mean, it should be self-evident, really, but it bears emphasizing. Molecular structure and function are inseparable. And if this becomes very, very important, for example, if there's any mutation in the DNA and a molecule deviates from the normal, because very often even a very slight change in the structure of a molecule can radically alter its function. Usually not to a good end. That's usually a bad thing. Occasionally it may be a good thing, and that may feed the evolution of a species. Molecular structure and function are inseparable. So here is a word which you have probably heard before, but it has a strict definition that we need to understand. And that is, macromolecules are most often, not all the time, in 3 of the 4 classes that we are going to talk about, they are very, very commonly, they are polymers. And that is, they are very big molecules which are made of repeating smaller molecules. And the polymer will be the long finished molecule. The small building blocks we call monomers. And the 3 classes where we commonly see polymers, and often very large polymers, are the carbohydrates, proteins, and the nucleic acids, as I've already told you. Those are polymers, and very often extremely large polymers. The fourth group of macromolecules are the lipids, and they don't form polymers, as you'll see in a while. They can be very big, but they don't do it by there are no lipid monomers to add together to make a lipid polymer. So the others, carbohydrates, proteins, nucleic acids, they have their own monomers. Each of those classes have their own monomers, as we'll see, and those monomers get added together in a specific order, that order determined ultimately by the DNA, which constructs a molecule of a particular sequence. That sequence has a particular form, and because it has that form, it has a particular function. So I want to just, you don't need to worry over much about the details of the chemistry. I just want to show you how these molecules can be built, and they can also be broken down. So if I want to add monomers together to make a polymer, here's how I do it. Here, we assume here we have a short polymer there, and we're going to add another monomer here. And to do that, we take out a water molecule, H2O. There it is there, H there, OH there, H there, OH there, and we take out H2O, and we end up joining this monomer to the longer polymer. So there's a mechanism, the mechanism to this is, for all of these polymers, is the monomers all have a small part which is either identical or very, very, very similar in all of the monomers. So they have this. If I was talking about a protein, for example, each one of these would be an amino acid. That's the monomer of proteins is an amino acid. All amino acids have one part of them which is identical. And they have the character of the amino acid is given by something, by a side group here. It's off here. We don't worry about it at the moment. The important thing is all amino acids, for example, have this bit which is identical. This is really important because it means that I can actually join these in any order. It means that any order is possible. The order is very, very, very carefully determined. It's determined ultimately by the DNA, by the genes. That is the genes that determine the sequence of amino acids. But because they're joined through identical parts, I could have another protein which read 312, for example, or 213 or whatever. There's nothing to stop you doing that because these are all identical. So, okay, so that's a dehydration reaction. We don't only make macromolecules, we also break macromolecules down. And there can be various reasons why that happens. Sometimes the protein's not needed anymore, it's broken down. Sometimes we break things down to construct another molecule. And when we come to the carbohydrates, we'll see that we often break molecules to extract the energy. This process here requires an input of energy, but that's really useful because the bonds here that are forged actually contain chemical energy and they contain potential energy. We can actually break this bond. And we do that in the cell by adding water. So we made the molecule with a dehydration reaction. We break it down by putting in a water molecule. It goes back to where it was. But what happens when we do this is that there's some energy released. And that energy can be harnessed by the cell and it can be used. So these polymers are very often not proteins so much, where they can be used for fuel. But we're going to see the carbohydrates, especially the carbohydrate polymers, they are a very, very, very important store of energy for the cell. That's our major energy store, is in carbohydrates. This process here is called hydrolysis. So we made the polymer with dehydration. We break the polymer with hydrolysis by adding water. There's a huge diversity of macromolecules inside any cell. And as I re-emphasize, the incredibly diverse range of polymers can be built from very few monomers, because the monomers can be joined together in any order. So, for example, all of our nucleic acids, DNA, RNA, as we'll hear later, are formed from just 5 monomers. And in the nucleic acids, remember this, in nucleic acids, those monomers are called the nucleotides. All proteins in the cell are made from only 20 amino acid monomers. And the proteins are unbelievably diverse. They perform a huge range of functions. Many, many, many structural proteins, all sorts of thousands upon thousands of proteins go to make up the cell and to drive its metabolism. And they're all made from just 20 amino acids. Why? Because we can join the amino acids together in any order, that order determined by the DNA. So let's have a look at our first class of polymers, of macromolecules. I'll say they're not always polymers, but our first class of macromolecules, and these are the carbohydrates. That word carbohydrate tells you exactly how they are made up. They are made up of carbon, a carbon atom, and a water atom. Carbohydrate. That's the carbohydrates consist of carbon, hydrogen, and oxygen. There are some very simple ones which we do make use of, and these are the monosaccharides. That's a simple unit. We regard it as being the monomer, but these are single sugars. They may be complex, by the way. They can have quite a complex structure, but the monosaccharides are the carbohydrate monomers, and we can add those monomers together. We can start to make polymers, and those polymers are common in the cell. We'll see in a minute. I just want to emphasize, that the carbohydrate monomer is the very first molecule which is manufactured by plants during the process of photosynthesis. That's the first complete biomolecule which is manufactured by plants. And so glucose is very important throughout the living world. It is very frequently used as a fuel source, but it's also used for many other things to build macromolecules, as you'll see in a second. So here, the monosaccharides, for the most part, they have a chemical formula. It's CH2O, 1 carbon, 1 molecule of water, CH2O. So if we go to glucose, for example, C6H12O6, divide this by 6, CH2O. So that's the basic recipe for a carbohydrate monomer. There are some that differ slightly if they have strange bonds, but in general, that is the formula for a monosaccharide. There are lots of them. The other one you may have heard of is fructose. It's also a plant product. Galactose, ribose, these are all monomers. You don't need to remember the names. There's only one I want you to remember, and that is ribose. Just put that in your head, ribose, because we're going to talk about ribose when we talk about nucleic acids. It's a sugar which is part of the nucleic acids like DNA. So these monosaccharides can act as fuel for cells all on their own. Glucose is a very good source of energy for the cell. And It is one of our major sources of energy. But these monomers can also be put to other purposes, and the most common purpose, other than fuel for the cell, the most common purpose is in plants, where they are structural, and we'll see that in a second. So again, a disaccharide is our shortest polymer using carbohydrates made from 2 monosaccharides. Here we're going to see one. Here's glucose there. This is fructose, and it's a very, very, very similar molecule. It has an extra carbon on it. But this, you can see this. This is the monomer. Here's the other monomer, glucose, fructose. Here's the water molecule we're going to remove in a dehydration reaction. HOH, H2O, and there it goes. And that joins them together into this little disaccharide. It's only got 2 sugars, so it's a disaccharide, one that you know very, very well, because this is sucrose, and that is common table sugar. Table sugar consists of 1 glucose and 1 fructose molecule joined together. We can carry on doing that. We can carry on adding monosaccharides one after the other, and when we do that, we start to create other compounds. And these are the polysaccharides. Once you get past disaccharide, you start to think about polysaccharides. And here is an instance where structure and function are intimately tied to one another, because a slight change in the orientation of the monomers can radically alter the whole nature of the polysaccharide. So the first one we're going to talk about is important because this is the major plant storage polysaccharide, and it is starch. And starch is a very stable polysaccharide. It's actually largely insoluble, and it consists entirely of glucose. It's all glucose molecules all joined together in a particular orientation. In plants, it occurs in various forms. The simplest one is amylose. You don't need to remember that. Just be aware. Starch itself can have slightly different forms. It can be branched or straight, have just a linear joining of the molecules. Because it's insoluble, it starts to precipitate out. It's made, this is a chloroplast. This is a little organelle that performs the action of photosynthesis. And so then inside the chloroplast, the plant will make glucose from photosynthesis and immediately start, if the cell doesn't need fuel, doesn't need glucose, at that time it will start putting the glucose together into a form of a starch molecule, and that will form grains inside the chloroplast. Eventually, this chloroplast will be packed with starch, and it then just forms a little storage. The photosynthesis stops, and it forms a storage organelle. It's packed with starch. And the plant cell, by the way, has a gazillion chloroplasts. So as some of them get filled up with starch and stop working, there are plenty others being made. This is starch. It's a polymer. The monomers in starch are glucose molecules all joined together by dehydration reactions. Now, that's in plants. In animals, we don't have starch, but we have a very, very closely related substance that is our storage molecule, our major storage molecule for energy which is readily available to the cell. We have other storage molecules, as I'll tell you in a minute, for longer term storage. But the storage molecule which we have, which is made up purely of glucose monomers, is called glycogen. And its structure is rather similar to starch. It's just highly branched. Here, as you can see here, it also precipitates out, and it forms granules inside our cells. We can actually see it. You can see it in bacteria as well. it's a common storage compound. And in animals, we don't have starch at all. We only have glycogen, and it's a polysaccharide. That is our storage polysaccharide. Interestingly, it is largely put into the liver. That's our major store of glycogen. And it's also packed into the muscles, because the muscles are always requiring quick energy. So rather than mobilize glycogen from the liver and transport it to the muscles, instead muscles have their own store of glycogen so they can grab energy quickly. And energy is derived by breaking these bonds between the glucose. So that's the first thing. And then you may also break up the glucose molecule itself and extract all the energy that's inside that glucose molecule. That's why it's a store of energy. So I told you the other thing about carbohydrates is they can be used to actually build material. And this is especially important in the plants, because plants can manipulate molecules, the glucose molecules. When they make a polysaccharide, they can make a polysaccharide called cellulose. Here's starch, as we have already seen. All that I want you to notice is look at where the OH groups are here. Okay, they're all on this side. This is starch. Glucose, 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 glucose. Well, here is cellulose, and it's also glucose, 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 glucose. But look at the orientation of the glucose. You got one in one orientation, the other one is flipped. Okay, you've got one in that orientation. The next one is in that orientation. Okay, so, and that simple difference of orientation of those glucose molecules makes this a radically different substance. Cellulose is an extremely strong molecule, insoluble, and it is the primary means whereby a plant builds and supports its structure. It forms the basic skeleton of plants. It is extremely common in plants. They are packed full of cellulose. It's very, very useful to plants to keep the body of the plant rigid against the forces of gravity. That's its major function. Can do all sorts of other things. Well, it can be protective, etc., etc. All plant cells are surrounded by a cellulose cell wall around them. And so that's its importance. So it does not occur in animals. Is not made in animals. But there's a major problem with cellulose in the biological world, and that is it's almost indigestible. Human beings and most animals like us cannot digest cellulose at all. It passes straight through your gut as digestive fiber. And it emerges exactly the same as it went in, it unchanged by our digestive processes. But that's a big problem ecologically, because if you think about it, plants make a huge amount of cellulose. And if the cellulose never decays, then a huge amount of carbon and everything would get wound up just in cellulose. And we can't have that system. Our molecules on our planet have to be recycled. Fortunately, there are some microbes, both bacteria, fungi, and also a few protozoa are able to digest cellulose. And they are responsible for breaking down plant cellulose and recycling it. Now you realize also that there are many, many animals which actually only eat plant matter. And if that cellulose could only be made available to them, it'd be a very, very valuable store of energy, of nutrient. But most animals cannot digest cellulose. So instead, animals like all of the things like horses, cows, goats, all of these animals that eat purely plant material. They have in their guts, they have these microbes which can digest the cellulose. They live together with these microbes. The microbes digest the cellulose in a safe, warm environment, and the animal derives the benefit of the products of the digested cellulose. A very complex process. This living together of an animal with another animal to both their benefits is a symbiotic relationship. That means living together. It's a symbiotic relationship. Cows and animals like that have a very complex gut. The stomach area of the gut is extremely complex. It has different chambers with different populations of bacteria and protozoa and all sorts of things in it. It's there specifically to digest plant material, to digest the cellulose and make it available. And by the way, that also means that cellulose enters all of those microbial digestions, mean the possibility of the components of cellulose re-entering the global cycles that feed atoms to living things. Okay, so let's now those are the carbohydrates. Now let's look at the next class, and these are the hydrocarbons. And carbohydrate was carbon plus water. Hydrocarbon is hydrogen alone with carbon. So it's just hydrogen and carbon. And that gives those hydrocarbons, they're extremely important in living things because these, for two reasons. The first is, you remember I told you that each cell is described by a cell membrane. That membrane is made of hydrocarbons. Secondly, hydrocarbons are our major long-term storage mechanism and are often created as oils or fats for a living thing to have a long-term storage of energy. Very often they will build up lipids and fats when times are good, put away, and then it can be accessed when times get tough. So the oils and fats are the typical hydrocarbons in biology, and they have one characteristic in common, all of them. It distinguishes them from every other macromolecule as well, and that is they are hydrophobic. That means they repel water. Hydrophobic means they repel water. The opposite is hydrophilic. If something is hydrophilic, it dissolves very easily. And that becomes important when we look at a particular class of hydrocarbon. If you think back to those carbon skeletons, these hydrocarbons are often extremely long skeletons of carbon atoms joined one after the other. And then with all these hydrogens attached to them, they have many chemical bonds. And each of those chemical bonds can be accessed for energy. So these are very good stores of energy because they've got so many chemical bonds. And that is usually their major thing. So here's one. This is a fat. Fats are large molecules, and they tend to solidify at quite low temperatures. Very often at room temperature, a fat will solidify. You don't need to worry over much about the structure, but I'll just point out, just have a look here. These black triangles are carbon atoms. The white are hydrogen. Okay, so here you can see long hydrocarbon chains, and fats often have three. And the fat is three of these joined together here with a glycerol. That's a sugar molecule. It's another sugar, and that's glycerol there joining these three together. That's our in human beings. That's our commonest lipid, and it is stored. This is a single, here's a diagram. Here's a cell which is specialized for storing fat. So here's the nucleus of the cell there. And you see all of these here, they're all fat droplets. They have their own little membrane around them. That's an organelle, which is specialized for storing fat. And these collections of cells form visible masses of fat. And they can also be, obviously the fat is there for energy purposes. It can eventually be accessed. Once we've laid down fat, actually it's very difficult to access it. It's only accessed when times are really tough. But often as well, we will lay down fat, these masses of fat, as protection for organs. For example, the kidneys are capped with a big cap of fat cells, which is protective for them, as well as being an energy source. So the lipids do not form polymers. There are no lipid monomers to join together. We see there are three important groups. I've already shown you the fats, and I'll show you again now. The other group are the phospholipids, and the last group that we're going to talk about are the steroids. So it's fats, phospholipids, and steroids that we are interested in. Fats I've already shown you, that glycerol molecule with the 3 fatty acids attached to it. Fatty acid, because here's a long hydrocarbon chain, but look at the end here, and that group there, all you need to know about it, it's acid. It makes this slightly acidic. That's why this is called a fatty acid. And we join them the same way we did before. We take out a water molecule and join here, and we end up forming the fat molecule in that way. I'll just tell you, because you may notice if you go to the doctor for your checkup and they do blood work on you, they will report the concentration of triglycerides in your blood. It's a measure, it's one of the health measures that is looked at. If the triglycerides are too high in your blood, it's not a good thing. In most people, that is not a problem. Okay, so again, there is this association between structure and function. And the fats are very variable. There are many of them. There are many different kinds of fats which are manufactured by the cell. And there are just 2 that I'm going to point out to you. And these are, first of all, those fats like this. You can see every single carbon here has as many hydrogens as could possibly be attached. So those we call the saturated fats. And in the saturated fats, the fatty acids are straight. Saturated fats have a characteristic that they tend to be solid at room temperature. And in addition, saturated fats are associated more strongly with adverse health outcomes. If you have a high intake of saturated fat, it is not a good thing. You want to avoid it. Saturated fats are predominantly, not entirely, but predominantly animal in origin. Things like cream, butter, those sorts of things, those are all saturated fats. And you just remember that they're solid at room temperature, because think of butter. It's solid at room temperature. Unsaturated fats tend to be plant-based in origin. And they're very often, their fatty acids associated with them, they have basically the same structure. Here it is, glycerol with the fatty acids associated. But often the fatty acids have double bonds in them. And if we were to, every time you double bond between 2 carbons, you have to sacrifice a hydrogen. So when you count it up, you see that not every carbon has every possible bond attached to a hydrogen. Instead, some of them have 2 bonds to the neighboring carbon. It does 2 things. It bends that molecule in space, but it also confers another property, and that is that they tend to be liquid at room temperature. If you want a typical example, olive oil. But you also know, if you go to the supermarket and the supermarket has their heating down, they got it really cool, you'll see that the olive oil actually does solidify at below room temperature, not far below room temperature. There's nothing wrong with it. Take it home and have it in a more warm environment. Normally at room temperature, olive oil is unsaturated. And unsaturated fats are not associated with adverse health outcomes. They are so-called healthy fats. Though probably if you had too many of them, it would also not be that good. So I just want to mention one other thing. So those olive oil and all of those sorts of things are called unsaturated fats because they don't have every single bond on the carbon with a hydrogen. Some of the bonds are to another carbon. They are unsaturated fats. But there's just one other class that I want to warn you about as far as health outcomes. This is now becoming increasingly uncommon. It's a group of fats called the trans fats. And these are artificial fats like margarine and various other things. The reason they were created is that they don't decay. All the other fats, the natural fats, they tend to oxidize very easily. And when they oxidize, they give food a nasty, rancid taste. Trans fats do not. They don't decay. And so they became very widely used, both for like frying, etc., but also in products, in biscuits and in donuts and in crackers, all of those sorts of things. And margarine is an example. The problem is trans fats are even worse than saturated fats for adverse health outcomes, especially cardiovascular. And the health outcomes are so drastic that in general, their use has been curtailed in food products. Rather put up with food going off than the person who eats it going off. The last one, the thing I need to tell you, especially for in human biology, the thing we need to understand, we are very good in many cases at taking basic components and manufacturing whatever fatty acids or fat, whatever we need. Our cells can manipulate these molecules quite well. But there's one group of lipids which we cannot manufacture. And so they are called essential fatty acids. We have to get them from our diet. Other things are able to manufacture them. Human beings cannot. So we have to get them from our diet. And these, typical example, are the omega-3 fats. And these you get from things like seafood, especially, very rich in seafood. But also, if you don't eat fish, you can also get it from nuts and grains as well. You just need to check. Things like walnuts, very, very high in omega-3 fats, and these are essential. There is some evidence that it's health-wise, it's very advantageous to make sure you have a high intake of omega-3s. There is some, not very strong, but there is some association, for example, with neurological health and omega-3 fats. Okay, so I think that's all I want to say about that section. I just wanted to now tell you a little bit about the last group of lipids that we need to study, and those are the phospholipids. Don't freak out at this diagram. You don't need to remember all of this. I just want to point out to you, this molecule is a very strange molecule. First of all, it looks like a little fat. It's not, it's actually a lipid, but it has these two fatty acid chains. There's glycerol, but attached to the glycerol, instead of a third fatty acid chain, attached to the glycerol, there is another group. And the only thing you need to worry about here is this here. And remember that this is a phosphate group. It has another group here as well, but this is a phosphate group. This is a strange molecule because, remember we said lipids are hydrophobic. That's strongly hydrophobic. It is repelled by water, or it repels water. This group here, however, is very, very hydrophilic. It likes water a lot. So this is schizophrenic molecule. It has a hydrophilic head and a hydrophobic tail. And it's going to behave in a peculiar way when I put it into water. And when you put them into water, they self-assemble into what is called a bilayer. And here it is here. Just look, you can see one phospholipid molecule. It has its hydrophilic head dissolved in water and a hydrophobic tail pointing in. And on the other side, there is another one, hydrophilic head, tail. It does this so that it can exclude water. It repels all the water in between. And it's perfectly happy like this because the heads are hydrophilic and they are dissolved in water. And the tails are hydrophobic and they are away from the water. But it's made this bilayer. And that is exactly the structure of the cell membrane. That is what makes up the skeleton of the cell membrane. So phospholipids are the major component of cell membranes. They're critically important to the functioning of the cell, to the structure and functioning of the cell. Just remember, they do this because they have, part of them is hydrophobic, part of them is hydrophilic. The last little group we're not going to discuss in any huge detail. I just want you to be aware that they are lipids. And these are the steroids. The reason that they are classed as lipids is because they are strongly hydrophobic, but their structure is completely different to the fatty acids and everything else. Instead, they are ring structures. They have ring structures. And they are all based on this lipid, cholesterol. You've heard bad things about cholesterol as far as health, and we'll talk about it later on. But cholesterol is essential because it forms the basis of many of our steroids. And the steroids that you probably heard of are things like testosterone and estrogen, things like that. The sex hormones are steroid hormones. Various other hormones are also steroids. The cholesterol has, by the way, has another really important function, and that is part of the cell membrane as well. But where we're going to encounter it again, we'll encounter cholesterol when we talk about nutrition, but we'll also encounter cholesterol when not cholesterol so much, but the steroids we will encounter when we talk about human hormones. Okay, so that is that for today. We'll complete this section hopefully then on Wednesday. And I wish you all well. I hope that you are well. And I hope that by now you have all sorted out your problems with accessing the Zoom classes. And I will see you then on Wednesday.